Good morning to everyone present here. I'm Gautham Dharuman. As JC mentioned, I'm a staff scientist at Argonne National Lab. I work with Arvind, Tom, Rick, and Ian. And today the topic is MProt-DPO. So this was the work that we presented at this past supercomputing conference. We were one of the 6 Gordon Bell finalists. So I'm happy to go over the details of the science, the scale and the software stack and the challenges we face. And then I think I will let Väinö take over to go into the details of the challenges and optimizations that he and others had to do for scaling it on the Aurora machine. All right, with that, MProt-DPO, the name stands for Multimodal Protein Design Workflow using direct preference optimization. We were successful in scaling this framework and crossing the ExaFLOPS barrier on 5 emerging exascale systems. Next slide. Next slide. Yeah. So today's to give an overview of what the science problem is. Today's efforts at protein design largely rely on sequences that have been sampled over the course of evolution. Techniques like directed evolution rely, in fact, on this principle of what nature has sampled and how that can be engineered to achieve new functions. But the combinatorial complexity of sampling a protein, say with just 100 residues, is enormously challenging because it leads to on the order of 20 raised to the power of 100 unique possibilities with a very small sample of that space that has been observed to achieve a particular function. This landscape of functional proteins defined by the sequence and structure space is usually referred to as the fitness landscape. And mimicking this process in the lab involves several steps that includes techniques for combinatorial optimization in a multidimensional setting and other variants of those approaches. So despite advances in high throughput characterization, as well as advances in generative methods, there is a challenge still as of today in characterizing protein fitness landscapes and optimizing their function for various purposes. Next slide, please. So let's look at the landscape of protein design tools that have been developed, exploiting similarities with natural language. Protein language models have evolved to come up with dreamt-up sequences that achieve certain class of function. And one of the key things that came out of these approaches is the need for building new representation for proteins. Next click. These representations have resulted in Nobel Peace Prize as recently as in 2024. However, an open challenge in the design of proteins that are tailored for specific function with the fitness landscape defined. Next slide. All right, so our motivation for approaching this particular problem was manifold, but one of the interesting differences that we brought into this application is a natural language interface for providing the user the ability to query sequences of certain design constraints, but using a natural language input, right? So we have heard talks on different foundation models for various domains. And if a foundation model is very specific to a domain, now we need prompting techniques and prompts that have to be specific to that domain. And that can be a limitation for a new user or a person from a different domain to be able to explore. Or in the case of experimental protein designers, um, it might still be cumbersome to know what exactly do the prompts need to be for certain class of models and what are the different bookkeeping that they need to do, right? So to overcome those limitations, one clean approach is to be able to provide a natural language interface to foundation models and give them the power to be able to query for specific design constraints just with a simple English description. So we are not the first, but the first model that came out in this paradigm is ESM3, which takes into account sequence structure functions as special tokens. But it is still very hard to prompt a model like ESM3 because of the way because of the way they thought about the design for it, and also uh, the functional keywords that you could input into ESM3 is still very limited in terms of um, not being able to specify quantitatively what exactly you want in the range of functions that are of interest. For example, um, what we want is a model. If you query the model and you ask, is there a way uh, to design a mostly hydrophobic protein with, say, 7 transmembrane alpha helices that can mediate cell signaling? Then the AI model should be able to generate something like on the right, which is a naturally occurring structure protein. And our hope is in this process of exploiting existing models, such as the llama suit, uh, by augmenting with knowledge about proteins, that we are able to have a very powerful model and interface for designing novel proteins across diverse families. Next slide, please. So why is this challenging? If you look at knowledge about proteins, they are captured in silos. That includes biochemical literature described in databases, as well as other sources like experiments and simulations that sort of live independent of each other and are distributed. Next click. One more. Thank you. Our hypothesis is that by exploiting intrinsic representations of this knowledge that is captured by transformer models, we can link these diverse representations across these modalities. Next click. However, incorporating simulations and experiments may not be straightforward with transform models, which means that the numerical representations need to be captured within the context of the fitness landscape that we are trying to optimize. Next click. Therefore, we use simulations and experiment data in the context of preferences that can be encoded to guide generative models, which capture various different aspects of the protein family of interest, ranging from GC content, catalytic activity, binding affinities, etc. And we would like to demonstrate this in the context of different applications such as enzyme redesign or antibody design, which has implications in development of therapeutics as well as design of catalysts for recycling. This, in summary, is our paper. Next slide, please. The key achievements that we have been able to do in this paper includes the first class of multimodal protein language models that can directly incorporate experiments or simulation data as feedback for protein design. So as I was mentioning, we have a natural language modality added, but uh, on top of that, we are also having an approach to incorporate feedback from experiments and simulations into the model so it doesn't remain static. And also this buys us other further advantages, like we can minimize hallucinations, we can choose more productive designs for experiments, and so on and so forth. We will demonstrate these in our results. From an algorithmic perspective, we designed and scaled multimodal protein language models that can interface with natural language while simultaneously scaling it on diverse exascale computing systems. This model is built on the best-in-class open-source Llama herd of models from Meta, that is keeping their backbone architecture while incorporating individual modalities for text and sequences. To incorporate experiment or simulation data, we developed two strategies of preference optimization and also scaled them across multiple exascale systems. These two advances enable us to achieve greater than 1 exaflop mixed precision sustained performance across these systems, and further enhanced by an integrated computational workflow that is supporting protein design with crucial implications in therapeutics and design of catalysts. Next slide, please. So how did we do this? Here, I'm giving an overview of the different blocks of our framework and the paper. So we needed ways to incorporate protein sequence data onto natural language descriptions, which allows us to compose the input data for fine-tuning existing models like Llama, and we will provide the rationale for choosing Llama backbone for fine-tuning. This covers the first two steps. Next, we tested these models by incorporating experimental measures of fitness for an essential protein in yeast called His7. And then these models are also used to guide generative AI for designing enzyme malate dehydrogenase, also known as MDH, using simulations as surrogates for catalytic activity using direct preference optimization. Next slide, please. The standard ways of representing proteins as sequences is great at learning co-evolutionary patterns, but they need specialized ways to prompt them, as I mentioned in my introductory slide. But on the other hand, natural language descriptions provide access to functional description of the proteins, but lack knowledge of the sequences. 2 clicks, please. Integration of the two can potentially overcome that problem. And that is by providing textbook-like description of every sequence in the dataset and their associated attributes, lifts the interface of design from just coevolutionary patterns to functional descriptions. But a similar template for representing all proteins would be less robust and lead to mode collapse. Therefore, we developed a mechanism to vary templates to diversify the representations and thereby preventing the potential issue of mode collapse from the dataset point of view. Next slide, please. This was also an interesting study that we did in terms of justifying the choice of our model sizes in a systematic manner, and we did the fine-tuning scaling experiments, and that's how we rationalized the choice of a model. And to just to not come across as being biased to just the llama suit, it's not about why llama versus the other models. It's about why we chose to fine-tune an existing backbone model, right? So that's what we want to give as a summary here. We can look at this by understanding the distribution of the number of tokens we have access to in our training corpus. Empirically, fine-tuning makes the most sense when the distribution of the number of tokens for achieving the expected loss follows the function that we show on the top right. 2 clicks, please. Oh, okay, I think you see the rest. This is dependent on the number of training examples versus the number of tokens and the parameters beta, largely. For the protein gym dataset, which is a corpus of experimentally measured sequences with just about 3,400 proteins, the descriptions of the protein sequences, their variants, and functions cover a total of 1.4 billion tokens. The total number of sequences was 2.75 experimentally measured and tagged protein sequences with their fitness measures. Um, but if you were to incorporate an entire world of non-redundant protein sequences from UniProt database along with their functional descriptions, that leads us to have anywhere between 150 billion to 300 billion. And if you get rich enough in your natural language description of the protein sequences, then you could almost have 1 trillion tokens in your dataset. And if you want to reach that particular loss, we require that many tokens, and our scaling studies indicate that as well, pretty well. Next slide, please. A second algorithmic innovation was in guiding the generation of our fine-tuned models via direct preference optimization strategy that we implemented in 2 distinct modes. The first mode is incorporating experimental feedback that encodes user preferences via a classifier trained on experimental fitness measurements. The second mode is a self-aligned one where the preference dataset is generated by the model itself that is getting aligned. The DPO loss function, which is sort of represented in a compact manner on the right part of the figure, is in fact perfectly formulated to achieve the above two intended feedback, both in uh, the conventional alignment and in the self-aligned mode, because the DPO loss preferentially weighs the preferred samples over the unpreferred ones to update the model weights. Since we have on the order of 2.7 million sequences in the preference data set, it was important for us to scale the DPO implementation within one of the most performance software frameworks for LLM training, which is Megatron DeepSpeed. The performance comes from fused kernels of NVIDIA's Megatron LM with zero optimization and pipeline parallelism of DeepSpeed, and flash attention to improve throughput of training. Moreover, since the Megatron DeepSpeed stack was originally tailored for pre-training and fine-tuning experiments, We had to incorporate novel code changes to implement two different data loaders for the preferred and unpreferred data streams. And further, since the DPO loss function is more involved than the simple negative log likelihood loss function in the pre-training or the fine-tuning mode, um, there were some changes that we had to make in terms to avoid the conflicts at the level of computational graph during the backpropagation phase, and the source for that was because if you carefully look at the loss function, there are two instances of the model that we have to keep at every instance of the training step. One always stays in no-grad mode, which is a reference model, and the other one is a model that's being aligned or tuned with our preferences. We trained on sequence lengths of the order 512 to 1024 for our target protein families. And you will see the more performances that the models achieve in the later slides. Next slide, please. So just to highlight schematically what the framework is, I'm sure Väinö will go into more details when he talks. But as I mentioned, so Megatron Deep Speed offers us this powerful combination of 3D parallelism combined with all the high-performance kernels. And so we, I'm not sure if when we did this work that we could have achieved the performance that I would be describing in the subsequent slides, if not for a framework of this capacity. Next slide, please. Also, to give a sense of the challenges that we had to face from implementing DPO, into the framework, I just wanted to have a schematic showing how it is different from implementing in something like Hugging Face, for example, which is pretty straightforward. But there are several nuts and bolts under the hood that goes on in the Megatron Deep Speed stack. For example, when you want to extract out the log probabilities, which are now fed into the loss function, you need to be able to carefully identify and trace that those are coming via the tensor parallel cross entropy terms, which further connect to how Megatron on its backend is computing those in the tensor and pipeline parallelism setting. And also unique to the loss function is once you get the log probabilities, now you are converting them into token-wise sequence log probabilities, and you're taking the ratios of the subsequent log probs from the model being tuned and the reference model for the preferred data set and the unpreferred data set via the two different data loaders that I mentioned. And finally, all of this is fed into a log sigmoid, and then you have a final mean over the batch to give you the final loss term that is now backpropagated uh, or via the different parallelism strategies for the model being fine-tuned. Right, next slide, please. So let me now proceed to discuss the results of our fine-tuning from the direct preference optimization experiments. Next slide. The first use case I'm going to talk about is an essential protein within yeast called His7. I'm told that this is a lethal protein. By that I mean, if you knocked out this protein, then the organism yeast would cease to exist. Also, it means that it's an important protein. So if there's a mechanism to really enhance the fitness of the yeast organism by optimizing the His7 protein, then you can imagine that it has very interesting applications all the way from brewing to cheese and whatnot. But that was not the motivation for choosing this particular protein as an example. In terms of the protein gym data set, this particular class of protein was also well characterized. So it was a very high quality preference data set that we could make use of. The enzyme His7 has about 220 residues and plays an essential role in the histidine biosynthesis. And just characterizing its fitness landscape involves a combinatorial explosion of 10 to the raise power 83, which by no means is possible to explore using experimental strategies. And as I mentioned in the first part of my talk, that even hypothetically, if there was a way to do that, it will not be useful because most of the space is sparse in terms of what are actually useful, feasible designs in the lab. And that's the other constraint to this current state. And one of the largest set of fitness characterizations was also done for this particular protein family in the data set by Pakistan and colleagues, and who segmented the protein into 12 independent segments and examined the space of 875,000 unique amino acid sequences that led to identify the fitness profile for the protein. So our goal was to succinctly capture this fitness landscape using the DPO strategy. The experimental data was thresholded to account for false discovery rates, and we used the same framework in the protein gym benchmark to build train, test, and validation data sets. Let's first look at the statistics of what a training data set looks like. So a large portion of this data represents unfit sequences, as you see on the bar plot on the right, um, and only about 25% of the data had fit experimental sequences. If we were to take the naive strategy of fine-tuning or supervised fine-tuning on either the sequence only or the text and sequence data, the resulting generations still mirror the same distributions as the training data, as you can see in the red and the green bar plots. However, let's take a look at what happens when we use the DPO method on the sequence-only model. Uh, two clicks, please. We get an increase of the fit sequences from about 25% in the training data to about 86%, which is more than 3 times increase. And if you include the text and sequence modalities together in the text sequence setting, there's a multimodal model. And if you did a DPO on top of that, then we increase the number of fit generations post DPO to about 91%, which is an extra 5% over the sequence only data with DPO. Which was very interesting because this intuitively means that the model has just learned something innate about this fitness landscape that allows us to reason about mutations in His7 that lead to favorable fitness properties. We then ask the question whether these properties are somehow encoded in the latent representations of the multimodal model that is getting tuned here. Next slide, please. So this is where we innovated in understanding where the attention was being placed in terms of the various layers across the network. We can visualize this by pointing to the separate tokens. And as you can see, when we select layers that have cross interactions between text and the sequence modalities, we can learn how the placement of the attention points to important residues in the protein. For example, when we place our pointer on the sequence composition token, it points to the dominant residues such as leucine, alanine, and threonine that constitute 36% of the composition. And this allows us to capture functional descriptions across the token space in this multimodal model. There are more complex interactions at the individual token level that may capture coevolution and/or functional descriptions such as aromaticity, isoelectric point, but we hope to learn more about this in our future studies. And this very amazing Wiz interface was created by Bharat, who is also a postdoc at Argonne. Next slide, please. Now let's turn our attention to the second use case, namely improving the activity of the enzyme malate dehydrogenase or MDH. Using a two-dimensional projection of the learned embeddings from the model for MDH, we see that the model embeddings capture the distribution of GC content for the underlying sequences. GC content is nothing but the number of Gs and Cs in the sequence, and that roughly correlates with the environmental survivability of the particular organism via the protein of interest. So each dot in this two-dimensional projection represents a sequence from the MDH training data. On the other hand, when we blindly generate sequences, we can get generations that follow the training data. We then generated 48 sequences from this space, highlighted by the white dots, randomly selected to start MD simulations that characterize the activation barrier for this enzyme, using a technique called empirical valence bound method, which uses weighted histogram analysis to model the potential of mean force involved in the hydride transfer step of this enzyme, which is considered to be the rate limiting step. We implemented this using the OpenMMD simulation framework for the paper, and it was mainly implemented by Väinö, who's also a staff scientist in our group. Using the barrier heights we estimated from the simulations that we then generated an additional batch of sequences using DPO. Next slide, please. The PMF profiles from the 5 wild-type sequences are shown in the gray here, with the best performing natural sequence having an estimated barrier of about 22 kilojoules per mole pre-clicks. And over the 2 iterations of DPO, we estimate a barrier height of about 18 kilojoules per mole, corresponding to a 3-fold increase in activity. A click, please. The open-fold predicted structures of the generated sequences are shown here with respect to the wild-type sequence. One click, which shows that these proteins also maintain the overall Roseman fold structure of the MDH. The binding sites for the cofactor and the substrate are also conserved despite the overall lower sequence homology. We now have about 48 variants that have been tested in the lab. Surprisingly, all of these have some MDH activity, but 13 sequences exhibit better MDH catalytic activity compared to the wild-type sequences. Next slide, please. So I've spoken to you about the algorithmic innovations and scientific results. Now I'll focus on the performance aspects of this model. We wanted to design, by design to execute this campaign on as many computing platforms as possible. The motivation for that is we wanted to present a framework that's not restricted to a certain hardware or network topology, but that is very generalizable to most of the state-of-the-art systems that we have out there, and we were able to do so for our final scaling runs. So our diverse computing platforms includes NVIDIA's PDX cloud machine, which has the H100 GPUs, the total of 3,200 for scaling runs. And then we had ALPS machine in Switzerland, which are the Grace Hopper GPUs with 4 GPUs per node spread across 2048 nodes. So there was a total of 8,240 GPUs for the scaling run. And then Leonardo was a total of 6,000 A100 GPUs spread over 1,500 nodes. And then Frontier has the AMD architecture with 4 GPUs per node. And finally, our highest number was from our own Aurora, which is the Intel Max 1550 GPUs with 6 GPUs per node. And we were able to scale it up to 19,200 GPUs. And I think that's what Väinö will go into greater details about the challenges that they had to overcome to achieve that scaling. Next slide, please. Next slide. So here, let me quickly go through our performance results in a nutshell. So for characterizing the 3.5B model at scale, so the training runs throughput is as shown on the plot. It summarizes the tokens per second, which is a measure of the throughput on each of the systems that we ran the model. This represents a weak scaling scenario for the fine-tuning runs. What you see from the plots is a linear scaling across all systems. One click. Let me draw attention to the performance at 256 nodes, where PDX achieves 35% improved performance over ALPS and 3 times better performance over Leonardo. Another click, please. At 1024 nodes, ALPS achieved 17% improved performance over Aurora, while ALPS and Aurora achieved similar performance at 2048 nodes. 2 more clicks, please. And Aurora's impressive performance is a consequence of leveraging the Rabin-Sniffer topology, aware-based collectives for performance at scale. So why are we thinking in terms of the throughput, right? So what does this enable in terms of pushing the science barrier or the frontiers in this particular application? As you may recall, the total number of tokens we expect for the full UniProt database, for example, with the rich textual descriptions can be anywhere between 150 billion to a trillion tokens. This means that the throughput achieved allows us to do rapid de novo design and connect it with ongoing experiments in autonomous laboratories at Argonne. In our runs, we used one rank per GPU with deep speed zero-stage optimizations, wherein the optimizer states are also shared across all ranks. And we chose the micro batch size to be as large as the GPU memory allows for each experiment. As we scaled the number of GPUs, we kept the micro batch size per GPU constant and scaled the global batch size accordingly. Next slide, please. Quickly, so this slide summarizes the scaling throughput for the 7B model. And as you can see, the runs were consistently linear scaling across all systems for both the models. One click. We see that the runs on Frontier at 1024 and 2048 nodes are sort of, on the bar with the runs on ALPS at 1024 nodes, which in particular achieved 35% improved performance over Frontier. Two more clicks, please. And at 256 nodes on PDX, we achieve a performance similar to 1024 nodes on Frontier. Next slide. All right, so coming to the punchline of what made us as one of the finalists and as one of the interesting submissions and one of the top contenders for the prizes, that all our scaling runs, as I mentioned, we were able to break the 1 mixed precision sustained performance on all the machines that I talked about. And by sustained performance, I mean that the measurement included the entire training iteration, including communication and I/O phases. We measured the flops using the deep speed flops file counter, which then was augmented to correctly account for the model parameters. And we carefully went through the arithmetic to make sure we counted every flop correctly, and that there was no overestimation of the ExaFLOP measurements. As you can see, the highest number was achieved on Aurora and scaled to 19,200 GPUs, giving us 4.11 exaflop mixed precision sustained performance. And the second runner-up was the run at scale on ALPS, but using all 8,240 Grace Hopper GPUs, and we reached 2.92 exaflop sustained performance on that machine. And once again, we achieved greater than 1 exaflop sustained performance on all the systems that we ran on. And as you can see, there's a very neat linear scaling as well for all the runs. Next slide, please. And yes, we just to show that the 7B model exaflop measurements were also equally impressive, and we achieved similar sustained performance for this model size with all the runs at full allocation scale exceeding 1 exaflop measurement. Next slide, please. So this table depicts the best performance achieved on various systems with another interesting metric that we report here in addition to the sustained or peak flops. And you can get the sense for the number of nodes and the number of GPUs that we ran at scale for our different campaigns. The key metric we use for performance is the percentage model flops utilization, or MFU, which is the ratio of the sustained performance to the rated max performance. As you can see from this table, we achieve MFU of 44.5% on Aurora and 48.4% on PDX. So PDX being a cloud machine, we were offered a total of 400 nodes with 8 H100 GPUs per node, so a total of 3,200 H100 GPUs that we ran the scaling runs on. But I think because of the way the stack and the network configs were quite optimized on PDX, it had a very impressive MFU numbers as well. And in terms of the peak performance, which illustrates the compute performance of the forward and backward passes, we achieved 5.57 exaflop on Aurora and 3.16 exaflop on ALPS. There were a lot of challenges that we had to overcome to achieve the scaling on full allocation scale on all the systems that we had access to. Primarily, many nickel timeouts, node failures, clever ways of handling the environment reads at scale. And I will leave with that summary here, and I will let Väinö take over to go into those details of the optimizations on Aurora and other machines here. Väinö, please take it over.
Thank you, Gautham. I wonder if we have any questions at this point, or should I switch out the slide deck?
Now, are there any questions because you'll move into more of the computational side of this project? I don't see any, so go ahead and start right now.
I hope you can see my slides. So I will go into the details of the computational side of things. So I will go into some details about the performance and software frameworks we used in this work. Then I'll quickly cover some other alternatives you could do on Aurora. And then I'll go into small details about the scaling runs we did. So what we had to do to get Python packages to load and initializing PyTorch, and what we had to do to optimize the collectives as well, just to get the nodes to communicate with each other. So first I have this same slide here. So demonstrating the near linear scaling on all the machines. So I thought I would start with this one. There's a slight dip with Aurora here at 1000 nodes. I think that was some kind of a mode switch for the communication library, so it didn't perform so well at that point, but we don't see this behavior anymore. I just thought that was quite an interesting detail here. But anyway, I think Gautham covered all the things about this one. So we used Megatron Deep Speed for this work. So it's combining the 3D parallelism of Megatron LM and has been augmented with Zero optimizer from Deep Speed. So Megatron LM has a lot of optimized fused kernels to really make it more efficient in terms of utilizing the GPUs. So for Aurora, there are ports of those kernels. So we are able to utilize those even though they were originally implemented for CUDA. And the same applies to the network stack. So originally, a lot of the PyTorch stuff is built on top of NCCL, but we were able to utilize one CCL for the Intel GPUs just as well. And also there was a mention of the flash attention too, which also we have on Aurora. So we were able to use that for more efficient transformer attention computation. And one detail here is that we had one rank per GPU tile. So even though in some of the earlier slides we had 6 GPUs on an Aurora node, each of those 6 GPUs has 2 tiles on it. And for this work, we used a tile as a device. So we actually had 12 independent processes per node, which is a performance detail, but that's how we recommend to do it for deep learning applications. Okay, and we used a fork of Megatron Deep Speed, like Gautham mentioned. We had some novel changes to the code to be able to do these things. So it was based on this Argonne leadership computing facility fork that you can find from the link. We have some minor changes here to the data loaders and such, just to make it work better for our use cases. And next, some deeper details. So there was a mention of using mixed precision. So why we went that way is because we have, well, for deep learning applications, you usually don't need the full precision of the single floating point numbers. So you can get near the same accuracy, but much faster computational throughput. So for example, I think the bfloat 16 is about 10 times faster on Aurora compared to the single precision floating point numbers. So you get 10 times the computational throughput with practically no loss for accuracy. But we had certain operations like accumulating the gradients and synchronizing them that already use in FP32 just for numerical stability, because those are a small fraction of the total compute. But this is why we talk about mixed precision, because some operations are still done in FP32 for stability. So like I mentioned, it's a tile as a device. So 6 cards, but 2 tiles per card, so 12 ranks per node. And we achieved over 100 teraflops per GPU tile, which meant about 1300 teraflops per node. I don't think for this work we used model parallelism for the final results, so the 3D parallelism pipeline tensor that was discussed earlier, but we have tested those features to work and we could have used them, but we ended up not needing them because the model sizes are not so massive that we would need to split the model across multiple devices. And we used the 1CCL communication library for optimized collectives. And it works the same way as NCCL. You just modify your PyTorch code a bit to use that instead. And like I mentioned, this was done with Megatron Deep Speed, but there's also other alternatives on Aurora, like using PyTorch FSDP to parallelize your deep learning workloads. And here on the slides, I have some links to our documentation in case you want to start doing these same things on your own. And there's multiple other libraries for fine-tuning and inference, and that we have tested, and the work is ongoing to support more of them. Okay, but next. So I would say next I will go in a bit more detail about the issues we had for running things at scale. So one detail is that the package management for Python packages, for example, if you use a tool like Conda or virtual environments or such, it can actually have tens of thousands of small files. And if you store that on a Lustre-based file system, you won't have much fun if you try to open it on tens of thousands of ranks simultaneously, because each one of those will do some metadata operations. So if you do hundreds of millions of metadata operations, your system admins probably don't like you very much, because we ended up making the whole network really slow for everyone running on the machine. So I don't recommend doing that. So what we did was just tarball the environment and broadcast it to the node local storage as a quick solution, because we had to still keep changing things, because I wanted to make some changes to the PyTorch code and so on. But the normal user should not really need to do that today, because, for example, our frameworks module is in the node image, so it does not load from the Lustre. And there's also this tool called Copper, which will do this free-like caching to speed up loading these huge software environments. So there's an illustration of Copper here on the right. So it's reducing the Lustre load by just having single rank in this image case, actually reading from the Lustre, doing those metadata operations. Then a second topic is just getting PyTorch to start at this scale. So what PyTorch does, it's an interesting design. So it opens a socket, TCP socket on rank 0, so the first process. And that process will have every other rank connect to it and exchange some simple information like how many of us are there and all that, like collect some counters and so on. But the issue is that when you have tens of thousands of processes connecting to a single threaded TCP socket, we didn't find it to perform so well. So for example, if you have tens of thousands of sockets, you need a return socket for each of those. And a socket is a file well, to simplify. So you need to increase the file count limits because the system did not allow to open so many return sockets. And there's also a kernel setting for limiting how many connections you can have in your socket queue. So if that limit is by default about 4,000, but we had 38,000 socket connections there. So it was possible to overload that and get connection rejected errors and so on. So we had to make some small hacks here and there to get PyTorch to be happy with that. But luckily today, that shouldn't be a problem anymore, because since PyTorch 2.4, they did some upgrades to it. So on the graph on the top right, you can see that there's significant improvement to the initialization time today. And I'm able to initialize PyTorch at any scale today with only configuring the socket number of return sockets. That's enough for that scale, but I still get some connection rejections. So I think the socket connections still need to be investigated, and that discussion is ongoing. But just if you ever want to run at this scale, these are the things you need to mind. And the next challenge is the collectives. So if you really have, for example, 4 gigabytes of parameters on tens of thousands of ranks, synchronizing those gradients with an all reduce starts to become a computer science problem. So for example, many of the communication libraries default to, for example, ring all reduce for the large scales, but that doesn't scale infinitely because it splits your data to so many chunks as you have ranks. But now we had 38,000 ranks, so it means 38 chunks sent 38,000 times. So that starts to suffer from the latency component there when you have tens of thousands of smaller sends. But luckily there, in one CCL, there's a lot of options for these algorithms. And we use the Raben-Seiffner's algorithm to get better performance at scale, because that does way less send operations with bigger packets. And we found that the network was working better for that. There was also a lot of environment variables we had to tune for one CCL and the underlying libfabric and the slingshot network to make those work as well as possible. But now we have those documented in the ALCF documentation. So if you want to do something like this, you can read about those. And of course, we are happy to discuss those with your application as well. And also, we've seen that this work led to some improvements in the whole network stack. So we had some challenges there at the high end, like on thousands of nodes, but today I'm able to do an all reduce at any scale without much changes, and I'm able to get a good bandwidth utilization of, let's say, 60% of the theoretical peak with 100,000 of ranks. So I would say that pushing these limits is important because today things work better. So as a summary, we achieved 4.11 ExaFLOPS on Aurora for sustained performance, linear scaling across all the systems. And we learned a lot about how to run PyTorch at the massive scales of tens of thousands of ranks. So we learned that, well, we already knew that storing Conda environments on Lustre is not really an option. So you need to do something else when you run at this scale. We learned that about the PyTorch initialization challenges, what the code does and what you need to do to make it better. But looks like someone else realized that as well, as PyTorch now has significant improvements. And we learned a lot about how to do large-scale collectives on Aurora. So now we have a set of optimized environment variables, and I think we are working on making that available as a module. So in the future, a user is just able to module load frameworks optimized, and it will set these environment variables automatically.